So before we move on to the ear and the special senses of equilibrium and hearing, we should discuss photoreception and how our receptors in the eye actually respond to light stimulation. So photoreceptors respond to photons. In physics for you, photons are really, really small packages of energy, radiant energy to be more specific, and they travel upon wavelengths. The distance between the peaks of the waves is determined by the color of the light. So, for example, photons of red light have the longest wavelength and the least energy, and photons of violet light have the shortest wavelength and the most energy. So rods respond to the presence or the absence of photons, regardless of wavelength. So they're very sensitive and very effective in dim light, but they don't provide us with any information about the color. That's where the cones come in. So we have three different types of cones. We have blue cones, green cones, and red cones. These cones contain pigments sensitive to blue, green, or red wavelengths of light. And since cones are less sensitive, they function only in very bright light. Color blindness can occur when one or more types of a cone is not functioning or if it's missing. The most common is a red-green color blindness where the red cones are missing. This is a great picture of a photoreceptor. So the pigment epithelium is the layer of the retina that borders the posterior cavity. It's the very top of this figure. The outer segment of the photoreceptor contains hundreds of thousands of flattened discs. And these contain visual pigments that absorb photons and initiate photoreception. The pigments are made of a compound rhodopsin that contains opsin and retinal. Retinal is derived from vitamin A. And retinal is the same in rods and cones, but opsin is different. The inner segment contains organelles and synapses with bipolar cells. Photoreception begins when a photon strikes rhodopsin. So if you look at the very top in the middle of this, top middle of this figure, you can see a photon as that yellow wiggly arrow. And it comes in and it strikes rhodopsin, which is represented by this big purple blob with the red bent key in it. Okay, so photoreception begins when a photon strikes rhodopsin. And the retinal and the opsin are going to break apart. And this is referred to as bleaching. This alters the rate of neurotransmitter release into a synapse with a bipolar cell. Okay. For a rod or a cone to be able to respond to light again, the opsin and the retinal must recombine. And so you can see that the retinal, the bent red key, once it is hit with a photon, it changes shape, and it no longer is bound to that rhodopsin. In fact, now you have just a retinal and an opsin. And it requires ATP to restore that retinal shape so that it looks like a bent key again, and to get it to bind to opsin again to form our rhodopsin. So it's kind of nice that this rhodopsin is rechargeable and recyclable, and that we can keep on seeing images. Otherwise, this would be a one-time event. The visual information received by the bipolar cell is then passed along to the ganglion cell. And the ganglion's axons form the optic nerves, or cranial nerve number 2. These are going to converge at the optic chiasma, and about half of the fibers cross the chiasm to the opposite side of the optic tract. The other half do not cross. The optic tracts then relay the visual information to the thalamic nuclei, which act as switching and processing centers, and send the sensation to the reflex centers of the superior colliculi at the midbrain, which controls eye reflexes, and or the visual cortex of the cerebrum. So that is the end of the eye and the sense of vision section of the textbook. The next section looks at the sense of equilibrium and hearing in the ear. So the special senses of equilibrium and hearing are provided by the ear, and the ear is divided into three anatomical divisions: the external ear, the middle ear, and the internal ear. The external ear is the visual portion of the ear that collects sound waves and funnels them towards the middle ear. The middle ear is a chamber with structures that amplify sound waves and then transfer those waves to the internal ear, which contains sensory organs for hearing as well as sensory organs for equilibrium. So let's take a look at that. The external ear is composed of the auricle, or the pinna, I think as your lab manual calls it. And this is a fleshy cup that directs sound waves into the ear and directly into the external acoustic meatus, or the auditory canal. This is going to contain many glands that are specific for secreting earwax. Those sound waves are going to strike the tympanic membrane, or the eardrum. The eardrum is a thin sheet that vibrates when the sound waves strike it. In the middle ear, which is also called the tympanic cavity, this is an air-filled chamber, and we have an auditory tube. This is also called a pharyngotympanic or an eustachian tube. But either way, this auditory tube leads to the pharynx, which allows for pressure equalization on either side of the eardrum. The middle ear also has three small bones that connect the tympanic membrane to the internal ear. So let's take a closer look at that middle ear and those three small bones. The first bone, the first auditory ossicle, which is attached to the eardrum, is the malleus. And the malleus vibrates on the incus, which is attached to the malleus and is the innermost middle bone. Sometimes this is called the anvil. And that is attached to the stapes. So the stapes has a base that nearly fills the oval window into the internal ear. Let's talk about that internal ear. The internal ear has sensory structures that are protected by a bony labyrinth. Inside of this bony labyrinth is a membranous labyrinth. And it's around this membranous labyrinth you have perilymph. So perilymph is a fluid that flows between the bony and the membranous labyrinth. And these tubes follow the contours of the bony labyrinth. And on the inside of that membranous labyrinth, you have endolymph. Okay, so you have perilymph around the membranous labyrinth and endolymph on the inside of it. So you can see that the oval window is covered with a thin membrane that separates the perilymph in the cochlea from the air in the middle ear. And just below that oval window, you can see the round window is labeled. And this is opening in the bone, which is covered with a membrane of the cochlea. So there are three parts of the bony labyrinth. The vestibule contains a membranous saccule and utricle with receptors for gravity and linear acceleration. The semicircular canals contain membranous semicircular ducts with receptors for rotational acceleration. And these two components, the vestibule and the semicircular canals, make up the vestibular complex. So this is going to provide us with a sense of balance. The third portion of the bony labyrinth is the cochlea, and this contains the membranous cochlear duct and the sensory receptors for hearing. So in the ear, we have hair cells, which are the sensory receptors. Hair cells are surrounded by supporting cells, and hair cells are going to synapse with dendrites of sensory neurons. The free surface of the hair cells is covered with stereocilia, which is where they got their name, hair cell. So it's the movement of this stereocilia that alters a neurotransmitter release. And so bending the stereocilia in one direction triggers depolarization. Bending of the stereocilia in the other direction leads to hyperpolarization. Now that you have an idea of the structures of the ear, let's discuss how it's used for the special senses of hearing and equilibrium. So there's two aspects of equilibrium, dynamic equilibrium and static equilibrium. Dynamic equilibrium is responsible for maintaining balance while in motion, and this is monitored by your semicircular ducts. Static equilibrium is maintaining balance and posture while you're motionless, while you're holding still. So this is monitored by the saccule and the utricle. The sensory receptors in the semicircular ducts are going to respond to rotational movements of the head. And so this is a figure that shows the three ducts, the anterior, posterior, and lateral ducts. And these are organized into three planes, transverse, frontal, and sagittal. Notice how each one contains an ampulla. Inside each ampulla, we have sensory receptors. The crista ampullaris contains hair cells that are embedded in gelatinous structure called the cupula. When the head rotates, endolymph pushes against the crista and activates those hair cells. In the vestibule, pictured here in this black box, the saccular receptors respond to gravity and linear acceleration. So the utricular receptors respond to horizontal acceleration. So hair cells of the saccule and the utricles are clustered in the oval maculae. The hair cells in the macula project into a gelatinous membrane. The surface of the membrane is covered with dense, heavy calcium carbonate crystals called otoliths. And so gravity pulls on these otoliths, pulling on the hair cells. Hair cells of the vestibule and the semicircular ducts synapse with neurons of the vestibular branch of cranial nerve number 8. Which then synapse with neurons in the vestibular nuclei of the pons and the medulla oblongata. And information is then relayed to the cerebrum, the cerebral cortex, the cerebellum, or motor nuclei in the brain, and the spinal cord. The hair cell receptors in the cochlear duct are similar to those of the vestibular complex. They provide us with the sense of hearing. They are responsible for conveying vibrations from the tympanic membrane to the oval window. The auditory ossicles are going to convert sound energy in the air to a pressure pulse in the perilymph of the cochlea. This is going to stimulate hair cells along the cochlear spiral. The pitch or the frequency is determined by which part of the cochlear duct is stimulated, whereas volume or intensity is determined by how many hair cells are activated at that site. So this is a sectional view of the cochlear duct, or the scala media, which shows two chambers. The scala vestibulae, or the vestibular duct, which is pictured on the top portion, and the scala tympani, or the tympanic duct. The scala vestibulae and the scala tympani are both filled with perilymph, and they're a continuous chamber. This is a closer look at the spiral organ of Corti, which is located in the cochlear duct on the basilar membrane. Hair cell stereocilia project onto the tectoral membrane, which is attached to the wall of the cochlear duct. So as waves strike the basilar membrane, moving it up and down, the hair cells are pushed against the tectoral membrane, bending the stereocilia. So the 6 steps to hearing are involved in this figure. In step number 1, sound waves are striking the tympanic membrane, causing it to vibrate. And in step 2, you can see that vibrations in the tympanic membrane are going to vibrate the auditory ossicles. These ossicle vibrations are going to lead to vibrations as pressure on the perilymph through the oval window in step 3. Step 4, the pressure pulses are going to distort the basilar membrane. And step 5, movement of that basilar membrane distorts the hair cells against the tectoral membrane. This leads to a neurotransmitter release. And then step 6, that neurotransmitter release that action potential is going to travel to the central nervous system through the vestibular cochlear nerve, which is cranial nerve number 8. So the cochlear branch of the vestibular cochlear nerve, cranial nerve 8, those axons arise from the spiral ganglion to the cochlear nuclei of the medulla oblongata, to the inferior colliculi of the midbrain, to the nucleus of the thalamus, to the auditory cortex of the temporal lobe. And that concludes the hearing and equilibrium section of chapter 9. I have a link to a hearing video that if you would like to watch it, you may. All you have to do is open up the PowerPoint file, which is in Blackboard in Learning Plans.